Okay. Um, Time to move on to chapter 4. Okay. All right. So this is where we ended yesterday. Okay. So okay. Now let's go over the different types of um, epithelium. Okay. All right. All right. So first one, simple squamous epithelium. One single layer of very, very flat cells. Okay, this is it's like a really, really thin piece of paper. It's not going to protect you from anything. Okay, what it is going to allow you to do is for things to pass through. Like it's easy to see through a single sheet of paper, so it's easy for stuff to move from one side of the paper to the other. So simple squamous epithelium really is for passage of materials by passive diffusion or filtration. Okay, and the two layers of the serosa, it secretes a little bit of lubricating solution, but you don't need to make a lot of it. You just need to make very little of it so you can kind of rub. So simple squamous epithelium, passive diffusion. Okay, where you will find it. Okay, your renal corpuscles, your linings, okay, of the heart, your alveoli. So let me show you what that looks like. Okay, so this is what this looks like. Okay, okay, single layer, very thin cells. See the huge amount of empty space? That's a really dead giveaway for simple squamous epithelium. Okay, all you want these things to do is to allow gas exchange from one side to the other. Okay, so this is where you'll find it. Okay, for all of these guys, I want you guys, so for all the pictures, I want you guys to understand what is it that makes this one like different than all the others. Because literally, I will give you guys a picture and ask you what type of epithelium or what type of connective tissue it is, etc., etc., etc. Okay, so be prepared. Okay. All right, simple cuboidal epithelium. Simple, one layer, cuboid, square-shaped cells. Okay, one layer, square-shaped cells. So here they are. Okay, so what you're looking at is the cuboidal cells around this this tiny little tube right here. Okay, this is um, a renal tubule. Okay, that is secreting stuff. And so it literally needs one single layer of very thin cells facing the hole. Okay, the hole is right here and right in the middle. And the layer around it is secreting things into that hole. Okay, simple cuboidal epithelium. Simple columnar epithelium, more or less the same thing. Okay, it's a little bit thicker. And okay. So you see that here. Okay, single layer of cells that are columnar. And you can tell they're columnar by just the shape of their nuclei. Their nuclei, even their nuclei are stretched out. They're no longer circular. Okay, so their function, absorption, secretion of mucus, enzymes, other substances. Okay, and here we have more stuff. Okay, these surfaces are called microvilli. Okay, we'll talk about what microvilli are, but microvilli are kind of important, okay? They help increase surface area, okay? And we'll talk about it later exactly how it does that, okay? But microvilli increases surface area, okay? So the ciliated form, in small bronchi, uterine tubes, the uterus, the non-ciliated form, digestive tract, gallbladder, ducts of some legs, okay? The other type of cell that you will see in this type of tissue are things called goblet cells, okay? Goblet cells secrete mucus, or better yet, they secrete mucin, which when you add to water, you get mucus, okay? Okay, pseudo-stratified columnar epithelium, okay? So all the cells originate at the basement membrane, okay? Only tall cells reach the apical surface, okay? So here is the pseudo-stratified columnar epithelium. And you can see that in my drawing earlier, yesterday. So you have, if you look, I can see one layer of nuclei here. I can see another layer here, and I can see a third layer there, okay? But if I look at each particular cell, every cell touches the bottom, okay? So I know it's not stratified, I know it's pseudo-stratified, okay? So pseudo-stratified columnar epithelium, okay? Again, the purpose of this secretion, particularly of mucus, okay, and it also helps move mucus along through things called cilia, okay? The cilia, okay, are much, much longer than microvilli, okay? The cilia actually generates current, okay? Let me explain what this does, okay? So here's your throat, okay? When you breathe air into your throat, the air is contaminated with a whole bunch of stuff, okay? And like pollen and bacteria and COVID-19 and other stuff that you do not want to be breathing in, okay? So, well, what do? Well, you have mucus, you secrete mucus. This stuff that you're breathing in will stick to the mucus. But having mucus full of bacteria is not a good thing either. So you have cilia that generate current. This current will move the fluid, the virus infected mucus up the throat and down the esophagus and into the stomach where you digest it, okay? And once you digest it, it really doesn't matter that much anymore, okay? Okay. So there. Okay. Tomorrow, sorry, stratified epithelia has two or more layers, okay? So two or more layers, they always regenerate from the bottom layer. The major role of stratified epithelia is protection, okay? And they're, again, like I said earlier, they're named according to the shape of cells on the apical surface. So let's look at the most common one, stratified squamous epithelium, okay? Stratified squamous epithelium have many layers of cells, okay, that are squamous in shape. Okay, so let's take a look at what these cells look like. Many, many layers of flat cells, and then the bottom layers are cuboidal or even columnar, mostly cuboidal. Okay. All right. Their thickness of the epithelium, and they're not meant for protection from abrasion. Okay, so rubbing. Okay, so you think your skin. This is this is your throat. Okay, so every time you swallow, the food scrapes along your throat as it goes down, and it's going to take cells with it. Okay, so it's not a big deal if you have like 30 or 40 layers here that you lose one or two. Okay, they can grow back. Okay, that is the whole purpose of stratified squamous epithelium. Okay, whether it's keratinized or not, okay, makes a difference. Keratinized, okay, forms the epidermis, the outer layer of the skin. The keratin kills the cells and makes it harder and drier. Okay, the cells on the surface of your skin are basically dead. They are dead. They have no nucleus. They have no function. They're just there for protection. Okay, keratinized epithelium are usually dead at an apical surface. Non-keratinized epithelium, okay, are going to be soft and mushy and alive at the apical surface. So your esophagus, mouth, anus, vagina in females, ureter in males. Okay, these are going to be non-keratinized, so they're going to be alive. Okay, so here they're going to be alive. So if you take a buccal swab, so buccal cheek, buccal swab is where they insert that Q-tip into the back of your mouth and rub, and you free off some skin cells, and then you look at them under a microscope. Okay, they have to do that. Okay, they have to do that to get off those cells so you can that you can swallow. Okay, so when you swallow, like the reason why these guys are not keratinized is these cells are alive at the surface. So when they take DNA, they need to rub hard and get those cells, and they can extract DNA from those cells. They can't get DNA from your skin because that's all keratinized. Okay. All right. Stop right here. Hold on. Okay. Let's continue. Okay, stratified cuboidal and stratified columnar. These are not going to be on the test, okay? Because you don't see them in very many places, and really they function like simple cuboidal and simple columnar. They just have multiple layers, okay? So for a little bit more advanced protection, okay? What you got to make sure you guys understand is transitional. Okay, transitional epithelium has both the characteristics of stratified cuboidal and stratified squamous. Okay, and where you'll see this is your urinary bladder. Okay, your urinary bladder is the only thing that looks like this. Okay. Okay, see these cells on the very top here? Okay, they're kind of round and they're kind of on top of the, like, kind of cover over multiple cells on the surface here. Okay, so right now that bladder that I'm looking at has no urine in it, so it looks round. As the bladder fills, this layer will stretch out thin, and so when it stretches out thin, it looks like simple squamous or stratified squamous. Okay, so when you look at transitional epithelium, you'll notice the changes in the top layer. Okay, all right, moving on. Glands. Okay, there are two types of glands. There are endocrine glands and exocrine glands. Endo secretes to the inside, exo secretes to the outside. Okay, so exo to the outside, they need a duct. Okay, so if you have a duct, it secretes to the outside. Okay, to the epithelial surface. Remember, the inside of your stomach is the outside of the body. So things that secrete through the inside of your stomach is the exocrine gland. Okay, endocrine gland. Endo, you don't need a duct because right on the inside, you're right next to the biggest duct of all, which is your bloodstream. And so I can just dump it into my lymphatic system, or I can dump it into my bloodstream, and it can go right there. Okay. All right. So the only thing I really need you guys to understand about that is the difference between endocrine and exocrine glands. Okay. Okay. The lateral surface features, the cell junctions. Okay. The factors binding epithelial cells together. Adhesion proteins. Okay. Special cell junctions. Okay. So let's talk about tight junctions. Okay, tight junctions are found at the apical region of most epithelial tissue types. Okay, so here, okay, these are tight junctions. So let me try to explain how they work. Oops, crap. Okay, so sorry, I didn't realize I wasn't sharing my screen. Okay, so go back. Okay, for the ducts, the endocrine and exocrine glands, I want you to know the difference between endocrine and exocrine glands. Okay. Okay. The tight junctions. So let's show you what tight junctions actually do. Okay. So tight junctions. All right. So here's my cell A. And here's my cell B. And here's my, oops. That is my tiny little bacteria that's trying to get into my body and kill me. Well, where will it go? How will it get through? Because everywhere you're going to encounter cells. So if I was a bacteria, the most logical path I would go through is to take that path, right? I would try to wiggle myself in between two cells and get in. Now, these cells want to prevent that, okay? So what these cells are going to do is build something right at the apical surface that connects it. And if it connects it, okay, the virus won't be able to go through. It'll get stuck right there. Okay, that is what tight junctions are for. Okay, they act like little Ziploc bags, so they act the way a Ziploc, the Ziploc part of a Ziploc bag will act. Once they link, okay, you can't get through the edges. Okay. Okay. The next thing I want to talk about are desmosomes. Okay. Desmosomes are the main junctions for binding cells together. Okay. So these are desmosomes. They work kind of like rivets. Okay. So what they do, okay, if I wanted, this is easier to show in a class setting. If I wanted to kidnap one of you guys, it'd be rather easy to do. I could pick one of you guys up, carry you guys away, and for most of you guys, I'm strong enough to do that. Okay. But I can't do that if you guys are holding on to each other. Okay? If you guys all held on, all held hands, and I pulled on one of you guys, I would either not be able to move you, or I would move you as a whole group. Okay? Like I would move the whole class if I pulled hard enough, but most likely I wouldn't be able to move anybody. Okay? Same thing here. If I'm linked, if I'm holding on to other people, okay, if I'm holding on to other cells, then they can't tear me away. And if they can't tear me away, they can't tear. Okay, so I prevent the tissue from tearing. Okay, this is really important at the boundary of a tissue to prevent tearing. Okay. All right, gap junctions. Okay, gap junctions are little connections here. Okay. Okay. If I want to send a signal, okay. One second. Okay. Let's go back. Okay. So we'll skip the basal lamina. Okay. Okay. The apical surface features microvilli. Okay, like I said earlier, this is how increased surface area. So okay, here again is my cell. If I wanted to increase surface area, okay, so the more surface area, the faster I can absorb things into the cell, or the faster I secrete things out of the cell, but mostly absorb. Okay, so what I want to do to this, say by that line up top, and now I want to make that. Okay, much more surface area, much faster absorption. Okay, so the difference, like for example, the difference between like the good paper towels and the crappy paper towels is simply that. Okay, how many little bumps they have, the more they have, the faster they absorb. Okay, like the cheap shit you find at school and stuff, the cheap paper towels, they don't have surface area at all compared to like the Downy stuff or Bounty or whatever whatever paper towel you buy. Okay. All right. Okay, cilia. So this is cilia. Okay, so it generates current. Okay, so here's the generation of current. Okay. All right. Okay, so I'm gonna stop this talk right here. Part 2 will be all about connective tissue. Okay.